We are delighted and honored to have today with us in our Bar-Ilan University Vision Science Seminar, Professor Morris Moscovitch from the University of Toronto. Professor Moscovitch was born in Romania and then at the age of 4 moved to Israel and at the age of 7 to Montreal, Canada. When he grew up, he did his first degree in psychology, graduating with honors at McGill University, where he became interested in memory research and did his undergraduate thesis with Peter Milner. This experience inspired him to seek a career in neuropsychology. He then continued to a master's and PhD in psychology at the University of Pennsylvania, where his thesis focused on hemispheric specialization and laterality in humans, and this was done under the supervision of Paul Rosin. During his postdoc year at the Montreal Neurological Institute, he worked under the supervision of the legendary Brenda Milner. Later on, he joined the University of Toronto and is there since. He is also a senior scientist at the Rotman Research Institute of Baycrest Center for Geriatric Care in North York that is affiliated with the University of Toronto. He did 2 sabbaticals at the Hebrew University and 2 at the University of Arizona, and these were instrumental in influencing his research and establishing long-term collaborations, especially with Shlomo Bentin of Blessed Memory and Lynn Nadel. Morris studies the cognitive neuroscience of memory, the changes it undergoes with time and experience from childhood to old age in neurotypicals and in people with brain damage or degeneration. He's been collaborating on this research with Gordon Winokur for many years now. He's also interested in how memory interacts with other functions such as attention, perception, problem solving, and prosocial behavior. And he also studies face recognition and hemispheric specialization. He has an H index of 140, according to Google Scholar, and over 72,000 citations. He published more than 350 scientific papers, many of them in top leading journals, including highly cited reviews, including in Ticks, in Nature Reviews Neuroscience, Current Opinion in Neurobiology, and many more. A few years ago, he was appointed a member of the Order of Canada for his contribution to the clinical neuropsychology and cognitive neuroscience, particularly his groundbreaking memory research. He received the William James Fellow Award, and please allow me to quote from the APS's website about his achievement. Well, through his elegant experimentation and brilliant theoretical insights, Morris Moscovitch's contribution to scientific psychology has ranged far and wide. Early in his career, his interest in brain function and cognition led to inventive research into the neural subsets of object and face recognition and hemispheric interaction and perceptual processing. This work provided the foundation of his component process theory, which stimulated renewed interest in the concept of modularity and functional localization within the brain. And I'm continuing. Morris may be best known for his contributions to the neuropsychology of memory and for his seminal experiments involving aged clinical populations that described the pattern of lost and spared memory function following different types of brain impairment. This work resulted in the influential multiple trace theory formulated with Lynn Nadel, which has reshaped current thinking about the function of the hippocampus and the ways the memories are presented in the brain. So without further ado, and even though there is so much more to say, I want to welcome you, Morris, to our seminar to hear about your exciting research, and we are delighted to have you today with us.
Thank you very much. I'd like to meet that guy. I just noticed somebody else on the screen, Jeremy. Hi, Jeremy. And Itzik Norman was there before, so hi, Itzik. Anyway, thank you very, very much. I'm going to speak a little bit out of my comfort zone. I usually do work on memory, and I still continue to do work on memory, but there's a series of experiments on the hippocampus and visual perception that I thought I'd like to talk about, given that this series is focused on on vision. So without going on any further, here goes. Whoops. Why is that moving? Let me just see how. I can't see my own screen is the problem.
Mm. The slide is passed to the next.
So the question is that I want to ask is, can understanding the internal organization of the hippocampus provide some clues to its contribution to visual perception and memory? So that's what I hope to achieve today. Be able to answer some of this question. But now the slide isn't moving.
I think you may be in annotate mode. Do you want to, I mean, because it looks like the pencil, maybe you want to turn off the annotation. I don't know, but
Okay, I don't know why. Okay, all right. Maybe I got into that
yeah.
by mistake. Okay, here. So here's two aspects of hippocampal organization and function that I want to talk about. One is specialization along the longitudinal axis of the hippocampus, and the other is pattern separation and completion. So part one, the longitudinal axis. So for those of you who are only interested in this part of the brain, the posterior visual cortex, this is what the hippocampus looks like and where it's situated. Here's another view of it. It's part of the parahippocampal gyrus in the temporal lobe and forms the floor of the inferior horn of the lateral ventricle and connects to the rest of the brain via the fornix, among other pathways. So this is what the hippocampus looks like, and you can see why it's aptly named the hippocampus, because it resembles a seahorse. Now, one of the things that I want to that figures prominently in my work on memory is that the functions of the head, body, and tail differ from each other, just as the functions of the head, body, and tail of the seahorse differ from each other. Now, this thing is pretty obvious, but it's only in the last 20 years or so that people have started focusing on differences of along the longitudinal axis of the hippocampus and see how it contributes to memory. So this is an MRI image of the longitudinal axis of the hippocampus over here. And I'll be referring to some aspects of this image through the talk. The first study that I'm going to talk about is a study conducted, led by Jordan Popping, who at the time was a graduate student at the University of Toronto and now is a professor, associate professor at Queen's University. And he had a problem. One of the things that he noticed in the literature is that the volume of the hippocampus, which one might expect to be related to memory, was not related to memory except in some unusual cases. And his insight was that people maybe are looking at the entire volume rather than trying to look at different aspects of the longitudinal axis. So he divided the volume into two parts. One part was the posterior neocortex, the posterior aspect of the longitudinal axis, starting at the uncus, being the demarcation point, and the other part is the anterior. The posterior connects polysynaptically with posterior neocortex. In short, it connects with the perceptual systems in posterior neocortex, particularly the visual system, whereas the anterior part connects directly to areas that have more to do with conceptual rather than perceptual aspects. The anterior temporal lobe, the ventromedial prefrontal cortex, and the amygdala that has to do with emotion. Once he did that, he found the very interesting, this just repeats what I just said, the connectivity of posterior hippocampus with perceptual posterior neocortex and the anterior hippocampus with anterior semantic or schematic neocortex. Here it is. Here's the division. When you measured the volume, what he found was something really quite interesting. The bigger the posterior hippocampus, the better the he looked at gathered data from a number of experiments where people looked either at recollection, and recollection, for those of you who are not in memory, refers to the ability to re-experience the past. So there are experiments in which participants are shown stimuli and then they have to comment on whether at retrieval they can actually re-experience having studied those stimuli in some detail. And there are a number of other markers of it, but I think that's sufficient. Often the recollection is accompanied by strong visual experience of having encoded the information. And what he saw was that the posterior hippocampus, the size of the posterior hippocampus, the volume, was related quite strongly to a person's ability to recollect the past, as opposed to merely recognize it as familiar. And the opposite holds for the anterior hippocampus, which is that the larger it is, the worse recollection is. Now, you might want to ask yourself why that should be the case. And that is that perhaps when people with large anterior hippocampi tend to focus on the conceptual aspects of the experience to the detriment of focusing on the perceptual aspects. And so you get poor recollection, though memory isn't poor as such, just recollection is. Okay, so here, along different firing and coding properties have been reported along the hippocampal long axis. If you look at rats, this is the rat hippocampus or rodent hippocampus. What you see is that there is a gradient going from the ventral aspects of it through the intermediate through the dorsal. In humans, you can see on this graph that the dorsal becomes the posterior hippocampus and the ventral, the homologue, is the anterior hippocampus. So we're trying to look at hippocampal place fields that were studied initially in rodents. Those of you who are not familiar with this, a place field is an area of an arena in which the rodent navigates that activates a particular region of the hippocampus. So those are place cells that have their receptive fields as place fields. So the hippocampal place cells were discovered by O'Keefe, and then O'Keefe and Adele wrote an initial paper on the functions of the hippocampus in 1984, and then a large book on the hippocampal organization and cognitive map in 1988, I think it is. So all these cells, the hippocampal place cells and entorhinal grid cells, code for the environment while the animal is navigating. Place cell activity, as you'll note, has also been observed in intracranial recordings in humans. So, for example, here's a rodent traversing an environment, and these are the place cells that will be activated as that rodent is traversing the environment. And you'll notice the size of the place cell is small in this case. But if you look at the ventral hippocampus, you'll see that the size of the place cells is much larger. There's a gradient, in other words, from the dorsal to the ventral hippocampus. And we wanted to see if we can get this representational gradient. Here it is depicted in another cartoon form. Evidence of this representational granularity in humans. So this is what I just mentioned. There's an increase in place field size from posterior to anterior hippocampus. And you could see that over here. Now, humans do have place cells, but let's think about some other aspect of it that may be the proper homologue to place cells in humans. There are viewpoint cells, that when you look at the world, there are view place cells, so that where your eyes are looking takes the place of the location in the environment that the rat navigates in. So these viewpoint cells in humans were found in the hippocampus. Now you'll notice the hippocampus is at the apex of the visual system. In some sense, it could be considered to be the terminal point of the visual system. And we wanted to ask the following question. Sorry, since many of the cells in the visual system have retinotopic representations, we wanted to see whether the hippocampus also has retinotopic representations, and whether they vary according to the way the place cells did, namely small retinotopic representations in posterior hippocampus, small representation with small receptive fields in posterior hippocampus, and large in the anterior hippocampus. So there's, as we all know, there's a contralateral hemifield bias in vision in the visual cortex. And this hemifield bias also shows up in the hippocampus. How did we do this? Or how was this done? Sorry, this thing is a little larger than I expected it to be. So there's in the Human Connectome project, there's a retinotopy part to it, in which individuals are shown while they're lying in the scanner with this field consisting of various stimuli the system is sensitive to. And retinotomy is measured by running this gap through this field, or looking at circles of whether it's activated by this region or this one, be indicative of a large receptive field, this a small receptive field, or a grid or a line grid that's moving across this visual field. And what you do is, what they did is measure the perceptual receptive field, or that's what we did. You can look at activations in the visual system that are retinotopically sensitive, which they have done, but we then looked at the same activations of retinotopic sensitivity in the hippocampus. And indeed, what we found was that there is a contralateral bias such that regions in the right hippocampus are activated more by voxels in the right hippocampus are activated more by stimulation of the left visual field, and vice versa for the left hippocampus. Now, what we measure are population receptive fields. We're not measuring single unit activity, although that may be possible, but we didn't do that. I seem to have, excuse me for just a minute. I seem to have downloaded the wrong slideshow.
Okay, no worries.
Let me just stop for a second and reorient myself.
Sure.
I was wondering why everything was so big. Let me stop sharing for a minute.
Sure.
Okay. So what we started doing is looking at population receptive field, which is the region of the visual field that will activate particular voxel or population of neurons. Yeah, this is where I was last time. Okay. There are two parts to this thing. In the Human Connectome project, there are two aspects of it. One was the retinopathy experiment that I showed you, and the other had to do with high-level visual cortex specialization that they inferred from NBAC tasks, where subjects, the NBAC task, of those of you who are not familiar, is a task in which you're shown a particular stimulus, and then a series of stimuli. They have to say whether you'd seen the stimulus NBAC, one back, two back, or three or four back. And the stimuli that they used were scenes, faces, objects, and body parts. And what we see when we look at it in the hippocampus is that the size of the population receptive field is strongly related to scene specialization. Hello. As compared to face specialization or face processing. So for here, for example, this is the difference, the advantage for processing scenes compared to faces. And you can see that the population receptive fields that are dealing with scenes are much more activated in the anterior hippocampus than the posterior hippocampus. And here you can see the size of the population receptive field receding, decreasing from the anterior to the posterior hippocampus. And what we found is that the relation of preference for processing scenes in the anterior hippocampus is strongly related to the size of the population receptive fields that we measure. So the population receptive fields show a nice gradient from anterior to posterior, much as a receptive field as spatial cells show in the rat hippocampus. But here, it also is strongly related, this receptive field organization along the long axis of the hippocampus is related to the kinds of processes that the different regions of the hippocampus specialize in. Let me just. So the next series of experiments that we wanted to see is whether we can find some measure of this overlap in the size of the receptive fields, or in the activations that go along from anterior to dorsal hippocampus. So the idea here is that these cells in the posterior hippocampus will not have much overlap from one to the other. Whereas as you move more anteriorly and the size of the receptive field grows, the overlap will be much greater. So how do we measure that? This is a study conducted by Eva Brunek, Boudica Belana, Morgan Berens, Jason Azupko, and us, and me. So here what we're doing is measuring the variations in the bold signal along the long axis of the hippocampus. And we could look at the temporal autocorrelation, how variations in one at one point in time overlap with activation at another point in time. That gives us the temporal autocorrelation. And we also wanted to look at intervoxel similarity, how much one voxel resembles how much activation in one voxel is linked to activation in an adjacent voxel. And that gives us a measure of intervoxel similarity and temporal autocorrelation. And we did this in resting state, and we also did it while participants were navigating Toronto in a visual reality task. Now, they could be navigating either without really navigating, actually, just following the arrow along an unfamiliar street, or trying to navigate in a familiar area, but one that isn't as familiar to them as other parts of Toronto. So you can have them navigating along a familiar route, an unfamiliar route in Toronto, or a mirrored route in Toronto where things are reversed, mirror reversed. And we wanted to see how these different types of navigation will affect the temporal autocorrelation and intervoxel autocorrelation. And we did this by using mass split into anterior and posterior segments along the long axis. I think there were 6 segments in all. And what we found is that temporal autocorrelation was higher in the anterior hippocampus than in the posterior hippocampus, as we predicted, because the overlap is fields are a lot larger here, and the overlap is going to be greater. So the temporal autocorrelation is higher in the anterior than the posterior. And so is the intervoxel correlation. Then we wanted to look at intervoxel similarity and navigation strategy that the people used while they were navigating through this virtual reality. And as you can see, the intervoxel similarity is highly correlated with the with navigational strategy, particularly for the posterior hippocampus. And it's also related in the posterior hippocampus with navigational efficiency. So we can see now that the posterior hippocampus, the kind of gradients that we see in the posterior hippocampus, are strongly related to participants' ability to navigate well. By the way, I didn't say this clearly enough or emphasize this. By navigational strategy, we had a test that distinguished between people who used cognitive maps and people who did not. And the navigational strategy is the proclivity for individuals to use a strategy based on cognitive maps that's related to the intervoxel similarity that's observed in the posterior hippocampus, as is the navigational efficiency, how quickly they go through that task and how few mistakes they make while they're navigating. Okay, this is just a summary of what I just said. Performance and taste of spatial memory, navigational efficiency, and navigational strategy correlate with the activity in the posterior hippocampus. There are also changes in cortical connectivity of posterior and anterior hippocampus with age. One of the typical things when we see in memory with aging is that older adults may have a reduced ability to remember details of to recollect or re-experience the past very well. they're conceptual. They can retain the gist of what they experienced, but the details are absent. So what you might expect is that there'd be some disconnection or loss of connectivity of the posterior hippocampus with posterior neocortex in these individuals, but connectivity of the anterior hippocampus to parts of cortex that are concerned with semantics and schematic information should be retained with age. And indeed, that we see as subjects age, they tend to rely more on connections between the anterior hippocampus and the ventromedial prefrontal cortex that's associated with schematic representations. Conversely, there's a significant decrease of posterior hippocampus connectivity of posterior hippocampus to posterior neocortex, consistent with what I just said about older adults' memory. So internal and external organization of the hippocampus indicate that posterior hippocampus supports fine-grained perceptual representations, and anterior hippocampus supports coarse-grained, just like schematic representations. Maybe it's good to just stop for a minute here and take questions if anybody has any.
Sure. I see that Christopher Tyler, he wrote a question in the chat. I can read it. So Chris is asking, what did the temporal autocorrelation measure that varied along the length of the hippocampus?
What did they measure? They
Yeah.
measured the activity, the activity that's correlated with. So maybe I should do it. Explain this in the part. Take a voxel in the hippocampus, and you measure how its activity is correlated with activity in the same voxel at a slightly later time point. So you measure the activity of a set of voxels in the posterior and in the hippocampus with themselves at a later time point. So it's measuring, essentially, another way of thinking about it is how long does that activity last in a particular set of voxels so that it'll correlate with itself at a later time point. I hope that's clear. If it isn't,
I also, is it okay that I also ask a question, or do you prefer?
no, go ahead.
So Eleanor McGuire had a study with taxi drivers in London where she examined the structure of the hippocampus and how that relates to navigation experience in the along the how long they have been driving. And I think she found that there was that she did look at the tail and the body, and she found that there was it's very similar to your results, where she found that the correlation was opposite to the years of or to the navigational skills. And I wonder if so I will look into that. But so do you think the posterior hippocampus is related to vivid memory? I mean, I'm just thinking because you're talking about navigation and also you mentioned earlier studies.
Sure. No. In fact, Eleanor McGuire did this quite a bit before we did our work. And indeed, what she found is that as your experience and as the experience of these taxi drivers increases, the size of the posterior hippocampus increases as well. And it's related to their expertise in being able to navigate in London.
Okay, wonderful. So it's
and in fact, she also found an interesting thing that the anterior hippocampus tends to be somewhat larger in bus drivers who drive along major routes rather than these little small streets in London that the taxi drivers will will drive in. So it's sort of related again, what we found in some of our own work on navigation that we did with Shana Rosenbaum. We found that indeed, what we think the difference is that along major routes, you don't need to have that precise visual representation of the route as you would when you're driving on the small side streets. And that's where the differences lie between um, the taxi drivers and bus drivers.
Yeah, okay, wonderful. Thank you.
Is that okay?
Yeah. Great.
Okay. So part one, conclusions. Evidence in humans of representational granularity along the long axis. Granularity changes with age, but in both young and older adults, granularity is correlated with performance on spatial and episodic memory. But what I wanted to emphasize, again, for the purposes of this talk, is that there's very strong visual representation, retinotopic representation in the hippocampus that may underlie the abilities, may underlie the function that the hippocampus serves in vision and navigation. Sorry. Again, I can't see my. So the second part has to do with studies on pattern separation and pattern completion. And the hippocampal subfields. One of the regions that we know is involved in pattern separation, seems most involved in pattern separation, is the dentate gyrus. And with Stevenson, Baker, and Shana Rosenbaum, we were able to study pattern separation in a patient with a lesion restricted to the dentate gyrus, where they found in another study that they had published that that individual had great difficulty in separating similar patterns in memory. But we wanted to look at now at categorical perception by looking at how people discriminate between morphs of faces. So we looked at faces of two famous individuals, Ryan Gosling and Benedict Cumberbatch. And we morphed them so that they go from morphs of 100% Ryan Gosling to only 60% Ryan Gosling and 40% Benedict, and all the way to the other end where it's mainly Benedict Cumberbatch and not Ryan Gosling. So we have a continuous series of faces. And we did that for famous faces, and we also did it for non-famous faces. And we looked at two things. We either had an identification task where some participants had to state what this morph, who this morph was of. They had to choose whether it was Ryan Gosling or Benedict Cumberbatch, or we had a discrimination phase where they shown two faces side by side, and they had to say whether they're the same or different. The idea behind this, we reasoned, is that categorical perception distinguishes clearly, allows you to distinguish clearly between a continuous physical dimension. I mean, the best example for that we can think of is the rainbow, where there's a, let's say, a change in hue that's continuous, but we see it as very distinct colors with very distinct boundaries. And in categorical perception, it means that you actually identify the stimulus as categorical, as being either Ryan Gosling or Benedict Cumberbatch. You don't identify it as somewhat in between. So we ask subjects to make these decisions and look at, this is, I think, Benedict Cumberbatch over here and Ryan Gosling up here, but it's a little small, I can't quite see, and ask people to make decision in the identification task. And what you can see, there's an ogival function, and that function has, let's say, a midpoint of where categorical perception occurs. So we can say over here, by and large, people see this individual as being primarily Benedict Cumberbatch, whereas over here below that point, they see that individual being primarily Ryan Gosling. Now, what we find is that for the blue lines are the control participants, and the red line is the person with dentate gyrus lesions. When we look at famous faces, the fact that the face is famous allows this individual to show categorical perception for these faces. But when the faces are not famous, categorical perception is impaired in these individuals. In this individual, this particular individual, he no longer shows that same nice ogival function, but more of a linear function tracking the differences in the physical aspect of it. And you see the same when we do a discrimination task, when you ask the person to discriminate between two faces that are shown on the screen and they differ from each other along a continuum. So when the orange is the patient with patient BL, and what you can see is that within category discrimination for famous faces is a little worse than controls. And the between category decisions is better. So if you're shown two faces that fall within a category boundary, participants are worse at it, and across boundaries, they're better at discriminating the two faces. So with famous faces, the patient BL looks a lot like controls. But when you get to non-famous faces, it's really striking that this individual cannot distinguish between two faces that fall within a category boundary along the continuum, but they're okay across category boundary. It's as if they don't have the pattern separation wherewithal to distinguish between small differences in faces that fall within a category boundary, but they can once the differences cross the boundary and are more pronounced. So damage to the dentate gyrus impairs pattern separation and categorical perception of faces. We haven't looked at other kinds of stimuli to see if it also applies to other stimuli. Incidentally, we did do this study and looked at inverted faces, and it breaks down with inverted faces because we think that categorical perception is not really appropriate for inverted faces. So now, last part of the talk is to look at pattern completion or integration. So now we're going to look at a comparison of HC, a person with developmental amnesia with atrophy restricted to the hippocampus and fornix bilaterally, but not to any of the other MTL structures. And again, we're going to look at face recognition. In this study, participants studied a series of faces in a fixed condition. So all the faces were in the same viewpoint. And then they were tested in the same viewpoint. And here it's again another fixed condition where they're shown faces, studied faces, all in the same viewpoint, but then were tested in a novel viewpoint. whereas they could study it variably, where they see faces, study a set of faces, each from a different viewpoint, and then they're tested in one of the viewpoints that they had studied, or they can study it variably again, but test it in a novel viewpoint. The idea behind this experiment is that in this case where the face is fixed, there's no need for any kind of integration of information across faces. It just sort of, you can just do it by straight stimulus matching, if you will, especially if you're tested with a viewpoint that you've already seen. Here you're shown this face is again in a fixed viewpoint, but when you go to a novel viewpoint, some integration of the facial information has to be accomplished so that you can modify it and recognize the face from a novel viewpoint. In the variable case, what you have to do is integrate all these different viewpoints so that you can form a representation of the face. And again, when you're shown a novel viewpoint, that requires you to apply that integration to yet another viewpoint of the face. When we look at eye tracking, this was done with Jen Ryan and Rosanna Olson in a study that looked at eye movements. And what's really interesting is that you can see one of the reasons that the patient, the hippocampally damaged patient, atrophied patient, is performing poorly on the integration task is that even while they're perceiving the face, the controls are integrating information from different aspects of the face, whereas the patient looks only at the eyes. And you can see that over here in the bottom. Let me get this up so that it's visible to you too. So you can see the patient looks a lot at the eyes, but almost at nothing else, preventing the patient to integrate the information across the face so that the face will be recognizable from different viewpoints. Now, if we look at the recognition performance, what we can see is that when it's repeated in the fixed condition, the patient does fine as long as the test is also in the fixed condition. But in the repeated condition, when the face is novel over here, okay, this is a stimulus that's repeated from the previous one. On all of these cases, the subject studied the faces in the fixed condition. They saw the same face over and over again, and then were tested with a repeated stimulus. HC shows a deficit, but only when they studied the faces in a variable condition. Let me repeat this just quickly. So these are controls in the fixed condition where they see the same face from the same viewpoint over and over again. This is HC in the fixed condition when he sees the same faces, the viewpoint over and over again. And the test is of a face that they had seen earlier. These are the controls. Sorry, let me go back. These are the controls in the variable condition, where they see faces from different viewpoints and then are tested with one of the faces that they had seen. And this is the patient looking at faces from variable viewpoints and tested with, again, one of the faces that they had seen. And you can see that the patient does fine in the fixed condition, but terribly in the repeated condition. Especially so if the face in the repeated condition is a novel face. In the variable condition is a novel face. So atrophy of hippocampus and fornix, output of CA1 impairs ability to integrate facial information, leads to deficits in face recognition that depends on such integration or when such integration is necessary. So the overall conclusion is that the internal organization of the hippocampus, let me see if I can get this, reflects its position at the apex of the visual cortex and helps account for hippocampal contribution to visual perception and memory. So just to put this into a larger perspective, here's a model that Robin Moscovitch and I published. It's now in 2017, where you have the anterior hippocampus. Because of the kind of representations that it has, connects closely to the ventromedial prefrontal cortex. The anterior hippocampus with large receptive fields in the anterior is able to get the general context of what the experience is like, and it's ideally suited to connect to information about schemas in ventromedial prefrontal cortex. And when you're recalling something, so in this case, what you have is just a schema that allows you to activate the proper gist in the anterior hippocampus in a memory test. And that anterior hippocampus then activates the posterior hippocampus so that you can elaborate on the perceptual details that allow you to show good recollection. I think that's it. Okay, thank you very much.
Morris, thank you so much. And I would like to ask everybody to unmute yourself and to give Morris a big applaud for really interesting and really interesting talk. And is it okay for you to take questions? Are
Yes,
you
yes, I'm happy to
yeah?
take questions. So it's nice to see some friends and acquaintances.
So okay, I'll start. I want to ask, I mean, you were talking about, which is quite novel to me, the idea that there is some coarse retinopathy, let's say right-left retinopathy, that is reminiscent retinopathy in the hippocampus. And I wanted to ask, I mean, it's also been found that there is some kind of reminiscent retinopathy in high-level visual cortex, but also, so I want to ask if there's also, you think, ventral and dorsal, like up and down aspects. I mean, that would maybe that would not be along the longitudinal axis, but I wonder if you think that would also be the case, or you only think that it's right, left, or yeah, this is
No, we also found retinopathy in looking up and down. Both of those, but not necessarily in what I thought you might be getting at, whether there's some differences in retinopathy as you move, let's say, along the visual cortex and the dorsal stream and ventral stream. And we haven't looked at that to know for sure.
Yeah, no, I was actually referring to the visual field and not to the
Okay,
specialization.
no, in the visual field, no, we get up and down, you get the same
Wow.
Get the same retinopathy in the hippocampus.
Wow, that's
Or
amazing.
looking up and down, and I didn't have a chance to say this, but we also get differences in eccentricity along the hippocampus, so that the eccentricity seems to be larger as you move towards the anterior than the posterior hippocampus. And you see the same kinds of things. This is picking up, I should just mention this. All this stuff on population receptive fields was inspired by a talk I heard Kalanit Grill-Spector give in Israel on population receptive fields. And I thought, oh my goodness, I didn't know you could do this kind of stuff. Maybe I can get some graduate students to look at population receptive fields. in the
Yeah.
hippocampus. And we were actually very happily surprised that it, not surprised, uh, uh, reassured that in fact you do see that same kind of retinotopic representation. And you see it also in the parahippocampus, primarily, again, the size of the population receptive fields in the anterior hippocampus is similar to the size of the receptive field in the parahippocampal cortex, parahippocampal place area, since both of them are concerned with scene perception, but in the perirhinal cortex, you get very small population receptive fields, consistent with its function and object perception, and maybe even aspects of face perception.
Yeah, I mean, thank you. It's exciting and it makes a lot of sense. I mean, center, high resolution, and of course, resolution with peripheral information. Jeremy, do you want to? Yeah, you're already unmuted.
Morris, that was interesting. So a bunch of us on the sort of the vision and attention side of the street have a lot of use for um, gist processing and the use of some sort of scene gist as a way to guide attention around a scene and things like that. And I was wondering whether your sort of frontal gist going through the anterior hippocampus, where on I'm looking at your picture because I took a picture of it where you're talking about generic cues to like house and party. Are you thinking of that as essentially semantic, basically giving you the word, or do you think that this pathway also has the capability to give you something, let's say, of the layout? Or is that would you think of that as living
Yeah,
elsewhere?
there are two things. Maybe just I'll just step back a bit. So what we think in that last little slide that I showed about gist and schemas, that what we think goes on when you encode information and then you're given cues to try to retrieve it. And the gist information that we think is represented in the anterior hippocampus is we didn't do really fine experiments to get at it, but in the memory domain, I don't know exactly how it would apply in the perceptual domain. In the memory domain, it would be being able to say sort of the series of events that may have happened in a particular episode without providing the peripheral details that accompanied that series of events. But in the perceptual domain, we haven't looked, aside from the fact that we looked at scenes versus faces, where we think that the anterior hippocampus having a larger representational field can actually give you the general layout of a location without telling you the real particulars of every aspect of it. So it's a coarse-grained representation of the visual world, is what I would have to say.
A different way of, or a different part of the same question would be if you're sensitive to the mirror reversal of your Toronto street, and I was pleased to say I was. How are you picking that up? Or where would that kind of grain of information be living?
What I was thinking about when you look at the mirror reversal of the Toronto streets and the posterior hippocampus there is the one that's most sensitive rather than the anterior hippocampus. The posterior hippocampus allows you to do that.
Okay.
Having been in that task, what I found myself looking at is very small details of the streets that I know, and you going from those details to try to figure out exactly where I was oriented in the street. So I'd recognize a particular building and say, okay, I know where that building is, and then refer to some. So that's what I think is going on in that. But it's not just like over there. Whereas the anterior hippocampus, I don't think it's capable of doing that. Or it doesn't have the wherewithal to be able to do that well, because it only cares about the general layout, which may be more or less similar, whether you see it in one and straightway or mirror reverse. Does that make sense to you, Jeremy?
Yeah. That sounds good.
There another question in the chat?
In the chat, no, I don't see another question, but I do want to ask, I mean, I know that the representation in the hippocampus, or at least to the best of my knowledge, is not as let's say the visual cortex has a very nice continuous map of the visual field, and I thought that the representation or the composition of the hippocampus is more sparse, so that adjacent neurons do not necessarily code similar things, but maybe I'm wrong on that. I mean, I'm, much less or much more distributed and not densely clustered together. So I wonder, do you think, so my question is, how does that fit if it's, I mean, do you think what you are sampling is some subpopulation of the hippocampus that has some retinopathy, or do you think that the signals represent, the whole population, or
No, that's an excellent question. So here, you're right that if you look at place fields in the rat hippocampus, one place fields that are adjacent in the visual in the real world are not necessarily adjacent in the brain. At the same time, and this is something that I didn't have the presence of mind to ask your question. At the same time, you see the same kind of representation, both the autocorrelation and the intervoxel representations. If you look at along the long axis of the hippocampus of the rat, you see the size of the receptive field, the size of the place fields changing from anterior to posterior. So now it's the size of the field that changes, but not necessarily the location of where the item is in the receptive in the visual field. So now let me circle back to your question. Does it mean that you get this kind of retinotopic representation of the entire field in the hippocampus? I think the answer is no. But in the way that it does represent it is that those with small receptive fields tend to aggregate in the posterior, and those with large receptive fields tend to aggregate in the anterior hippocampus. But they may not necessarily be related to each other in terms of where the location actually is in the real world. Does that make sense?
Yeah, I mean, Mel,
Mel?
Yeah, I just wanted to follow up and ask, in your own studies and also in that meta-analysis that you presented of lots of studies, I wondered, have you separated systematically retinotopic representations from spatiotopic ones? In other words, there would be a confound if people are fixating between whether or not it's a representation of something on the left or right, or whether it's a representation of the left or right visual field. And
I think in the data that we received from the Human Connectome project, I don't think there's any separation. They're confounded.
Yeah. So what's your hunch? What's your guess? I mean, presumably a lot of the place cells would be spatiotopic to some degree, but I wondered what
Yes, no,
what
I think they'd be spatiotopic partly because somebody may be more knowledgeable in the audience than I am. But the place cells, their location is they're also influenced by the head direction cells of
right. Yes.
the animal. So I think you sort of try to align it to spatiotopic representations rather than retinotopic ones.
So it might be interesting to do a study like left and right hippocampus, where you have people fixate eccentrically either on the left or on the right, and see if you can whether things move retinotopically or whether they stay in place, being a spatiotopic representation.
Yes, no, I think that's a great idea. I'm not the one that's going to do this study, but I'll pass it on to somebody who I
Me
think
neither.
can. No, that's a terrific idea. I have a postdoc right now, Hannah Tarter-Stoll, who's looking at population receptive fields and the hippocampus and going to record single units for
Oh, really?
epileptic patients.
Well, that
And
would be that would be a real opportunity, right?
so that would be an opportunity for her to
Yeah.
do that. Her interests lie in attention and memory, but I'll mention that to her. Thank you, Mel.
Oh, well, thank you for a great talk. I found that really interesting. I didn't know a lot of that. That was really quite something.
Thanks. Itzik?
Yes, hi. Thank you for this fascinating talk. And yeah, I'm just curious to ask you this question because this gradient from very let's say specific and small let's say place field in the posterior hippocampus to the more abstract and bigger place field in the anterior. And then I'm just trying to think how we think about the label itself, like the verbal label itself, for example, a name that to attach to a face that is so important for us to increase our ability not to remember this face again and to recognize it. And this label, this string of syllables that we call a name, is in one sense is very specific and very like episodic like because it's one exemplar, but on the other sense it's like abstract in a way. So I'm just curious, like how do you put it on this gradient?
Great question. Just extrapolating from my own data, I don't have.
Yeah.
So let's assume that the name comes from or is represented in anterior temporal lobe, and then that would have to activate some region of the. Oh, and you want to get its particular representation of the face. I don't know. I'm just wondering whether this is a hippocampal function altogether. I mean, the hippocampus is going to be good at trying to form representations of faces or be required to form categorical representations of faces, as I've argued. But having formed them, it may also affect how faces are represented in other parts of the brain, such as the fusiform gyrus, fusiform face area. But I'm not sure. I mean, I don't think I want to have the hippocampus do the functions of everything, but it's a good question. And I don't know whether anybody's actually looked to see whether giving somebody a name and then having the person imagine what the face looks like very clearly, whether that would require the hippocampus or not.
Yeah. No, I just feel like once you have the name, it's easy. You have some way of an easier access to to reactivate this facial, both the facial configuration and everything, but also the entire concept. And but it could be something cortical and just
So one possibility is to take another tack is that what's stored in the hippocampus is the association between the name and the face.
Yes.
And then given the name, you can reactivate the face. But I don't know whether that, I don't know to what extent that information is there in the hippocampus, or they're just a kind of code that allows you to get that information from other parts of the brain. No, I just don't know the answer. Great
Okay,
question.
thank you.
I wish I had a whole career ahead of me.
You do, you do. There is there are two. I mean, Christopher Tyler wrote in the chat, but maybe it's already. I don't know if it's relevant because he wrote that it's the same as Mel's question, but I will read it anyway. How do you think about the transformation between the retinotopic localization in the hippocampus and the environmental speciotopy of the place cells? But I think this is what you already answered, if I'm not mistaken. And Russell Epstein, which is here. Oh, here you are.
Hi, Russell.
Hi. And he's also going to be giving the talk next week, which is exciting. We're like, yeah, continuous. So he, do you want to read it? Do you want me to read it?
Oh, I was just in response to Mel's question about the retinotopy versus spatiotopy. I was brought to mind it in our study where we looked at grid representations in the Toronto cortex. We found that they would rotate with the visual display, so they seem to be tied to the boundaries of the visual display, which would imply spatiotopy. And I'm trying to remember. I think that the Killian and Buffalo study that originally found like grid-like responses for the visual display. I think they might have had a similar manipulation, but I'm not remembering that. So, so I think that the idea that these things might be spatiotopic as opposed to retinotopic seems likely to me. I'm
Yeah.
wondering if anyone else has actually done that study.
Just trying to think of some of the work that oh, God, his name suddenly escaped me. Ed Rose did on viewpoint cells.
Yeah, I mean that would be consistent with the spatial view cell.
Spatiotopic,
Yeah,
right.
certainly. Yeah. So
Wonderful. Maybe we're gonna, I mean, maybe we'll be also lucky to hear about that next week, but we will.
no, different topic.
Okay, different topic.
Yeah.
But I also want to ask, but I assume you're already, I mean, time is up, but I just had a thought, you know, about the what you said about the grid cells and the fact that they are kind of like phase shifts of the grid across space. So you have the same grid, but it's so maybe the fact that we don't see retinotopy in the hippocampus is because there is there is kind of like there is coarse retinotopy, but it is kind of like shifted. So it's not necessarily the two adjacent cells are related to the same grid. They might have, I'm saying grid, you know, retinotopical grid, but it might be that one is mapping space here and another one is kind of like shift, but when you look at it, they're not continuous in space. They may be continuous in more remote, I don't know, I have to draw this. But I'm thinking it could be also like the shifts in the grid in the place cells and the it could be also retinotopic wise that there is some shifts in the retinotopies, that it's not you know a real precise map, but more like overlays of different maps that are phase shifted along multiple directions or spatiotopical shifts. But anyway, just so
Before I get off, let me just say hello to the people I recognize on the screen. Nurit, hi Nurit, and hi Talia. Nice to see you. I'm sorry I can't give this talk in person.
Good to see you anyways. Thank you for the beautiful talk.
Thank you.
Morris, I want to say you really thanks a lot for joining us and you know for bringing to us you know an exciting topic I wasn't even expecting. So and it was a real pleasure having you. And again, I want to thank you so much for joining us. And yeah, it's been a pleasure. And
A
thank you
pleasure for
everyone in
me.
next.
Yeah, it's the first time I've actually tried to put some of this stuff together. Usually I talk about a different topic. But anyway, I figured maybe you'll get people interested in the hippocampus and vision.
Yeah, it's great. I mean, I assume it's, yeah, I mean, the people found it as exciting as I did. And we have Russell here next week, so we will send more details through the
Good luck,
email.
Russell.
Thank you, Morris.
Thank you. This was great.
Thank you so
Thank
much.
you for inviting me. It was a
All
real
right.
pleasure
If
seeing
you have
you.
a minute, maybe
Pleasure.
to say, maybe I can ask another question.
I can stay on. Maybe other people want to get off.
I'll
Go
also
ahead,
stay
Holly.
because I also have a small question.
Do you want me to stop the recording or do you want to
And I don't care.
me
Sure, you
neither?
can stop, yeah.
Okay, so I'm stopping the recording.